2.5 billion years ago, something extraordinary was happening in Earth's oceans. Life had already existed for well over 1 billion years. Entire ecosystems filled the planet. The seas were alive with microscopic organisms. Yet, if you had stood on the shoreline of that ancient world, you would not have been able to breathe the air. There was no oxygen. In fact, oxygen was completely absent from Earth's atmosphere for nearly the first half of our planet's history. The sky was different, the oceans were different, life itself was different. And then something changed. Something alive stumbled upon a new way of surviving. What happened next would transform Earth more completely than any asteroid impact, supervolcano, or mass extinction that followed. The strangest part is that the organisms responsible had no idea what they were doing. For hundreds of millions of years, they produced a substance that never reached the atmosphere. The oceans swallowed it as quickly as it appeared. Then, eventually, the oceans could hold no more. When that substance finally escaped into the sky, it triggered one of the most devastating catastrophes in planetary history. The dominant life forms of Earth began to die. The climate collapsed. Ice spread across the planet for longer than all later ice ages combined. And yet, without that catastrophe, nothing breathing today would exist. The organisms responsible are still alive. They are still performing the same chemistry tonight that they performed billions of years ago, unaware that they reshaped an entire world. In this story, we're tracing one of the most important turning points in the history of life, from an oxygen-free Earth to the accident that changed the atmosphere forever. Now, let's begin. Imagine Earth's entire history compressed into a single day. Midnight marks the planet's formation, and the hours begin to pass across a world still taking shape. Mountains rise and erode, oceans gather, continents drift and change. Through all of that long planetary morning, the atmosphere remains entirely free of oxygen. Noon arrives before even the first trace of it begins to appear. For nearly half of Earth's existence, the air belonged to a chemistry that would feel profoundly unfamiliar today. The atmosphere was a dense mixture of methane, carbon dioxide, nitrogen, ammonia, and hydrogen. Oxygen had no meaningful presence within it. A person standing beneath that sky would not have recognized it as the world they know. In composition, it likely resembled the atmosphere surrounding Saturn's moon Titan more closely than the modern Earth. Every breath available to life came from chemical pathways that no longer dominate the planet. Above this landscape, the sun shone with only about 70 to 80% of its current brightness. Under modern conditions, that reduction would leave Earth locked in ice, yet the oceans remained liquid. Methane provided the difference. Produced continuously by microscopic organisms, it formed a powerful greenhouse blanket that trapped heat and held temperatures high enough for water to remain unfrozen. The climate was supported by a balance that had persisted for immense stretches of time. Life itself appeared surprisingly early. Evidence suggests microbial communities may have existed as far back as 3.8 billion years ago, within roughly 700 million years of Earth's formation. Every known organism from that distant age lived without oxygen. Their biology developed in its complete absence. Many would have found oxygen actively harmful, a reactive substance capable of disrupting the chemistry that sustained them. The appearance of the planet reflected this different chemistry. The sky was likely tinted orange and brown through reactions involving methane high in the atmosphere. The ocean carried colors of its own, influenced by large quantities of dissolved iron suspended throughout the water. No ozone layer filtered incoming ultraviolet radiation. Sunlight reached the surface with an intensity unknown on the modern Earth, pouring directly through the atmosphere without the protective shield that would eventually form much later. Beneath those waters, the seafloor hosted a world populated almost entirely by microbes. Hydrothermal vents released heat and dissolved minerals into the surrounding ocean. Iron, nickel, and other metals circulated through the water in concentrations far greater than those found today. Thin microbial mats spread across sediments and rocky surfaces. No fish crossed the currents. No shells accumulated along coastlines. No reefs, forests, or animal tracks interrupted the landscape. What makes this world especially striking is its stability. It was not a temporary stage awaiting replacement. For nearly 2 billion years, Earth functioned successfully under these conditions. Its ecosystems were established, productive, and enduring. The organisms inhabiting them were adapted to a planet without oxygen and had no reason to anticipate anything different. Along certain shallow coastlines, however, evolution was assembling a new possibility. Within communities of microscopic life, a biochemical mechanism was taking shape that had never existed anywhere on Earth before. Across the shorelines of that oxygen free world, a new lineage was beginning to spread. It did not arrive with any visible sign of importance. There were no large bodies, no dramatic structures, no feature that would have distinguished it from countless other microbial forms occupying the planet's waters. Yet around 2.7 billion years ago, these organisms carried within their cells a capability that would eventually alter the chemistry of Earth more deeply than any mountain range, ocean current, or volcanic province. They are known today as cyanobacteria. Their descendants still inhabit oceans, lakes, rivers, soils, and rocks across the planet. Viewed under a microscope, they appear modest and familiar, little more than tiny chains or clusters of cells. Their appearance conceals how unusual they were within the biological world they entered. Earlier photosynthetic organisms already existed. Sunlight had been harvested for energy long before cyanobacteria appeared. Those older forms relied on substances such as sulfur compounds or dissolved iron to provide the electrons required for photosynthesis. Their success therefore depended on local chemistry. Growth could continue only as long as those materials remained available. Entire ecosystems were tied to finite mineral supplies, distributed unevenly across the seafloor and coastal waters. Cyanobacteria escaped that limitation through an innovation that transformed the scale on which photosynthesis could operate. The source they tapped was water itself. Oceans covered roughly 70% of Earth's surface, placing an immense reservoir directly within reach. For the first time, a photosynthetic organism possessed access to a resource available almost everywhere sunlight touched the sea. The ecological consequences unfolded across countless generations. Populations expanded into environments that earlier photosynthetic organisms could not dominate. Coastal habitats became increasingly favorable. Growth was no longer constrained by the same narrow mineral bottlenecks that had shaped previous biological communities. A new path had opened, and cyanobacteria followed it across broad stretches of the ocean. Their colonies formed layered microbial mats that spread over sediments and rocky surfaces. One generation grew atop another, creating stacked communities that trapped particles drifting through the water. As layer accumulated upon layer, these living carpets gradually thickened. Over long spans of time, they produced structures large enough to be seen without magnification. Low mounds rising from shallow marine environments where sunlight remained abundant. Evidence of these organisms survives within some of the oldest rocks on Earth. In the Pilbara region of Western Australia, researchers have identified microfossils and chemical traces consistent with cyanobacterial life dating to roughly 2.7 billion years ago. independent studies using molecular clocks, which compare the evolutionary history of photosynthetic proteins across living organisms, arrive at remarkably similar estimates. Two very different lines of evidence converge upon the same chapter of deep time. Their significance, however, was never rooted in size, abundance, or geographic reach. Cyanobacteria were extraordinary because of a molecular process operating within every cell. It worked continuously, generation after generation, producing a substance with little immediate consequence to the organisms creating it. For hundreds of millions of years, that byproduct accumulated almost unnoticed within the wider story of the planet. The mechanism itself was unlike anything that had existed before, a biochemical innovation of unusual power and specificity. Understanding it means stepping into the chemistry that reshaped Earth and set the course for everything that followed. At the heart of the cyanobacterial story sat a chemical capability unlike anything that had existed before. Tiny cells drifting through shallow water had acquired the ability to capture sunlight and use its energy to pull apart one of the most abundant substances on Earth. That ability is known as oxygenic photosynthesis, and from it flowed every major transformation that followed. The process sounds simple when described from a distance. Sunlight enters the cell, water provides electrons, carbon dioxide supplies carbon, sugar is assembled, oxygen is released. Yet inside that sequence lies one of the most demanding chemical achievements biology has ever performed. In its simplest form, six molecules of carbon dioxide combined with six molecules of water using light energy to produce one molecule of glucose and six molecules of oxygen. The glucose stored energy for future use. The oxygen drifted away as waste. That discarded oxygen often receives most of the attention because of what it eventually did to the planet. From the perspective of the cell producing it, however, oxygen had no value. The real prize was the sugar. The oxygen was merely the leftover material from a reaction whose purpose was energy capture and survival. Every cyanobacterial cell released it without any awareness of its future consequences. The machinery responsible for this reaction is known as photosystem 2. Researchers at the Massachusetts Institute of Technology and many other institutions have described it as one of the most consequential biological innovations ever to appear on Earth. The reason becomes clearer when examining the task it performs. Water molecules hold their electrons tightly. Removing those electrons requires a substantial input of energy. For many years, scientists questioned whether living systems could carry out such a reaction efficiently. Photosystem 2 solved that challenge through an arrangement of proteins and metal clusters capable of directing energy from sunlight into precisely controlled chemical work. As light entered the system, electrons were stripped from water molecules. The water was broken apart. Hydrogen participated in the construction of energy-rich compounds. Oxygen atoms remained behind. Pairs of oxygen atoms joined together and departed the cell as oxygen gas. Access to water changed everything about the ecological possibilities available to cyanobacteria. Earlier photosynthetic organisms depended on materials such as sulfur compounds or dissolved iron. Their productivity remained tied to local supplies. Water covered most of the planet. A photosynthetic organism using water gained access to a resource available almost everywhere sunlight reached. Natural selection favored that advantage generation after generation. No foresight guided the process. Evolution acted only on immediate survival and reproduction. Cells able to exploit water more effectively left more descendants. Those descendants inherited the same chemistry and spread into additional habitats. Evidence suggests photosystem 2 evolved only once. Every plant, every algae, and every oxygen producing photosynthetic lineage alive today traces its ancestry back to that single innovation. The proteins involved appear to represent a rare merger between previously separate systems, requiring an arrangement of extraordinary precision. Some researchers view that rarity as a reason oxygen rich planets may be uncommon across the universe. For hundreds of millions of years after this innovation emerged, the atmosphere showed no sign of it. The cells continued their work each day, releasing oxygen molecule after oxygen molecule into the surrounding water. Something else was intercepting that oxygen long before it could ever reach the sky. Picture a world where oxygen was being produced every day, in every season, across countless stretches of sunlit water, yet none of it could reach the sky. For nearly 500 million years, cyanobacteria released oxygen into their surroundings, and the atmosphere remained unchanged. The explanation was waiting in the ocean itself. The seas of that distant Earth contained dissolved iron in quantities almost impossible to imagine today. Modern seawater holds only trace amounts, but the early ocean carried vast reservoirs of it. Iron drifted through the water column, supplied continuously by hydrothermal activity and interactions between seawater and the planet's crust. Whenever oxygen appeared, the encounter was immediate. Oxygen combined with dissolved iron, producing iron oxide. That newly formed material became insoluble and sank toward the seafloor. In effect, the ocean behaved like an immense chemical filter. Oxygen molecules emerging from cyanobacterial communities rarely traveled far before meeting dissolved iron. The reaction removed them from circulation and locked them into mineral form. Generation after generation of microbes continued their work, while the atmosphere above remained almost completely untouched by what was happening below. The evidence for this process survives in stone. Across multiple continents, geologists have uncovered formations composed of alternating layers of iron-rich rock and lighter bands of silica-rich material known as chert. These structures, called banded iron formations, preserve one of the longest environmental records on Earth. When cut open, they resemble pages in a book written across hundreds of millions of years. Each layer reflects a recurring cycle. During periods when cyanobacterial populations flourished, oxygen production increased. More dissolved iron was converted into rust-like minerals and removed from seawater. As local iron supplies became depleted, iron-poor layers accumulated. Hydrothermal systems gradually replenished the ocean with fresh, dissolved iron, restoring the conditions for another phase of iron deposition. The sequence repeated again and again, creating the striking banded patterns visible today. Another line of evidence comes from sulfur chemistry. In rocks older than approximately 2.43 billion years, scientists find a distinctive arrangement of sulfur isotopes produced when ultraviolet radiation interacts with sulfur compounds in an atmosphere lacking oxygen. That pattern appears consistently throughout older sediments. Then it disappears. Once oxygen began accumulating in the atmosphere, the chemistry responsible for that signature could no longer operate. The change appears worldwide, creating one of the clearest boundaries in the geological record. The locations preserving these records span much of the planet. Western Australia contains the Pilbara and Hamersley regions. Comparable deposits occur in South Africa, Brazil, and the Iron Ranges of Michigan and Minnesota. Together, they supply much of the iron ore used by modern civilization. Roughly 60% of known mineable iron reserves trace back to these formations. There is a strange continuity in that fact. The steel supporting skyscrapers, bridges, ships, railways, and cities began as oxygen captured by dissolved iron long before animals existed. What industry extracts today is largely the accumulated residue of microbial waste preserved across immense spans of time. Even so, the iron trap does not account for the entire delay. Oxygen was being removed efficiently, yet another influence was operating alongside it, one that affected the cyanobacteria themselves and limited how much oxygen they could produce in the first place. Along the margins of the early continents, where sunlight reached the sea floor and tides moved across broad coastal flats, a peculiar architecture was taking shape. It did not arise from plants, animals, or any organism recognizable to modern eyes. Layer upon layer, microbial communities built structures that slowly rose from the sediment, creating some of the earliest large-scale biological landscapes on Earth. These formations are known as stromatolites. Fossil examples extend back roughly 3.5 billion years, making them among the oldest visible traces of life ever discovered. They appeared more than a billion years before oxygen accumulated in the atmosphere, preserving evidence from a chapter of planetary history when life remained microscopic yet had already begun reshaping its surroundings. A stromatolite was never a single organism. Each one consisted of countless generations of microbes living together in layered mats. Cyanobacteria occupied the upper surfaces where sunlight was available. Sediment drifting through shallow water became trapped across those mats. Fresh microbial growth spread over the newly deposited particles, forming another living layer. The process repeated continuously. Growth proceeded at a pace comparable to a human fingernail, yet over thousands of years, these communities produced structures reaching several meters in height, which is about several yards. Across the Archean and early Proterozoic coastlines, stromatolites expanded into extensive reef-like fields. No forests covered the land. No animals moved through the shallows. No shells, burrows, or coral formations existed. In many regions, stromatolite communities represented the dominant large structures visible along the shoreline. Their rounded forms stretched across tidal environments, creating broad biological provinces maintained by sunlight, water, and microbial persistence. Within those layered mats, oxygenic photosynthesis continued day after day. Oxygen escaped from cyanobacterial cells into the surrounding seawater, where dissolved iron remained abundant. The reaction between the two occurred rapidly. Iron oxide particles formed and settled downward, adding material to the growing deposits accumulating on the seafloor. Every cycle of microbial growth contributed another increment to the gradual transformation already underway within the oceans. Their influence extended far beyond their size. A single stromatolite might appear modest when viewed in isolation, yet vast populations operated simultaneously across coastlines around the globe. Collectively, these microbial ecosystems processed enormous quantities of carbon dioxide and water through photosynthesis, steadily releasing oxygen into environments prepared to capture it. The chemical consequences accumulated over spans of time, difficult to comprehend on a human scale. Living stromatolites survive today in only a handful of locations. One of the best known lies within Hamelin Pool in Shark Bay, Western Australia. The waters there contain salt concentrations roughly twice those of ordinary seawater. Such conditions exclude many grazing organisms that would otherwise consume microbial mats. Along the shoreline, modern stromatolites provide a rare glimpse into a biological strategy that first appeared billions of years ago and has persisted through nearly every chapter of Earth's history. The fossil record reveals that stromatolite abundance eventually reached a peak and then declined sharply around the onset of the Great Oxidation Event. Their story spans more than a billion years of uninterrupted construction. Layer by layer, generation after generation, they helped load the oceans with iron oxide while contributing to a planetary transformation none of their builders could perceive. At the same time, another chemical constraint was operating throughout the oceans, limiting cyanobacterial expansion and extending the long delay before oxygen could finally escape into the atmosphere. The delay in Earth's oxygen story was shaped by more than what happened after oxygen left the cell. The ocean also influenced how many cyanobacteria could exist in the first place. Even with sunlight above them and water around them, their populations were held back by the chemistry of the sea itself, as if the planet had placed a hand on the scale before the atmosphere could change. One of the strongest constraints came from nickel. In the early ocean, nickel was far more abundant than it is today, delivered by volcanic rock from a younger, hotter Earth. Much of the seafloor was made from ultramafic material, rich in minerals that released nickel into seawater. At high concentrations, that metal interfered with cyanobacterial metabolism. Cells could still photosynthesize, but their growth slowed. Colonies remained smaller than sunlight and water might otherwise have allowed. Nickel also supported another group of microbes whose influence reached into the atmosphere. Methanogens, organisms that produced methane, depended on nickel as an important nutrient. Their enzymes used it while generating the methane that helped keep Earth warm beneath the dimmer young Sunday. The same metal that restrained cyanobacteria helped sustain the microbial world that preserved the old greenhouse climate. Ocean chemistry, therefore, favored the oxygen-free world from two directions at once. Across hundreds of millions of years, Earth itself began to alter that balance. The interior cooled, volcanic activity changed character. The seafloor gradually shifted away from the most nickel-rich compositions. Less nickel entered the ocean with each geological adjustment, and the concentration in seawater declined. The change unfolded through mantle cooling, crustal evolution, and the replacement of one volcanic regime by another. With every reduction, the biological brake on cyanobacteria eased a little further. Urea added another layer to the restraint. In the chemistry of early seawater, urea helped shape the balance among microbial communities, influencing which organisms flourished and which remained limited. As ocean conditions evolved toward the early Proterozoic world, that chemical influence weakened. The microbial field became less weighted against cyanobacterial expansion. A population that had once grown under multiple constraints could now spread more effectively through sunlit waters. Laboratory experiments have tested these ideas by recreating aspects of early ocean chemistry. Researchers adjusted nickel and urea levels in controlled environments and measured how cyanobacteria responded. When nickel remained high, growth was suppressed. When nickel declined and urea conditions shifted toward later ocean chemistry, cyanobacterial populations expanded more readily. The pattern supports a view of the long oxygen delay as both chemical and biological. That finding reaches beyond Earth's past. A planet with oceans rich in nickel might host photosynthetic life for immense spans of time without ever building an oxygen atmosphere. Its microbes could be active, abundant, and productive, while every telescope, looking from afar, would see no oxygen signal at all. Life could be present, yet chemically masked by the world that contained it. On Earth, the trap and brake weakened together. Iron had been absorbing the oxygen already produced, while nickel had limited how much the bacteria could make. When both restraints finally loosened, cyanobacteria entered a new phase of expansion, and the chemistry of the planet began moving toward a threshold it had never crossed before. There are moments in planetary history when change arrives gradually for ages and then becomes visible all at once. Around 2.43 billion years ago, Earth crossed such a threshold. The forces that had restrained oxygen for hundreds of millions of years weakened together, and a world that had remained chemically stable for immense spans of time entered a new state. By this stage, nickel concentrations had fallen far below their earlier levels. The biological limits that once held cyanobacterial populations in check had eased considerably. At the same time, shallow coastal waters contained far less dissolved iron than before. Generation after generation of photosynthetic microbes had consumed the ocean's capacity to neutralize oxygen in many regions. The long established balance between production and removal was approaching its limit. Evidence preserved within the evolutionary history of cyanobacteria points to a striking development during this interval. Researchers, using molecular clock analyses, have identified a period of rapid diversification centered on this same window of time. New cyanobacterial lineages appeared in unusually large numbers, suggesting that ecological opportunities were expanding. Populations spread through surface waters more effectively than before, occupying habitats that had once remained beyond their reach. As those populations increased, oxygen production expanded across broader portions of the ocean. For hundreds of millions of years, the surrounding environment had absorbed nearly every molecule generated by photosynthesis. Now the remaining sinks struggled to keep pace. Oxygen began accumulating within surface waters in concentrations that earlier oceans could not sustain. The chemistry that had hidden it for so long was gradually overwhelmed. The geological record captures this transition with exceptional clarity. Among the most important markers is a sulfur isotope pattern known as mass-independent fractionation. This signal forms only under atmospheric conditions lacking oxygen. Rocks older than approximately 2.43 billion years preserve that pattern consistently across the globe. After this boundary, it disappears permanently. The change appears in ancient formations on multiple continents, providing one of the clearest chemical turning points in Earth's history. Another transformation became visible across the continents themselves. Red beds began appearing in the sedimentary record for the first time. These deposits formed when iron minerals reacted with oxygen in surface environments, staining rocks with reddish tones. Landscapes that had weathered under oxygen-free conditions entered a new chemical relationship with the atmosphere. The continents were beginning to rust in open air. Viewed against the immense delay that preceded it, the transition unfolded with surprising speed. After roughly 500 million years of oxygen production that left almost no atmospheric trace, oxygen shifted from near absence to a persistent and growing presence. On geological timescales, the change was abrupt. The cyanobacteria responsible for that transformation had not invented a new process. They had not suddenly become more sophisticated. Their cells continued performing the same reactions that earlier generations had carried out across countless ages. The difference lay entirely in the world surrounding them. The ocean could no longer absorb everything they produced, and the atmosphere finally became the destination for the excess. Once oxygen entered the sky in meaningful quantities, it encountered the gas that had dominated Earth's climate for nearly 2 billion years. The collision between those two atmospheric forces began almost immediately, and the consequences reached every corner of the planet. For nearly 2 billion years, a partnership between geology and microbiology helped keep Earth warm beneath a fainter Sunday. Deep within oxygen-free environments, Methanogens released methane continuously into the atmosphere. That gas accumulated over immense spans of time, creating a greenhouse effect far stronger than carbon dioxide. Without that atmospheric blanket, much of the planet's surface would have struggled to maintain liquid water. The arrival of oxygen introduced a new chemical relationship into that long standing balance. Methane and oxygen do not coexist easily. When they meet in the atmosphere, they react to form carbon dioxide and water vapor. In practical terms, every molecule of methane removed by oxygen was replaced with a greenhouse gas that trapped far less heat. A system that had supported planetary warmth for ages began losing efficiency molecule by molecule. At the same time, the source of that methane was coming under pressure. Methanogens evolved in environments completely lacking oxygen. Their metabolism depended on enzymes that functioned only under those conditions. As oxygen spread through more habitats, those biological systems became disrupted. Many methanogen populations declined, reducing methane production at the very moment atmospheric destruction of methane was accelerating. Supply and persistence were weakening together. Earth's climate responded to the change. Methane had carried a large share of the greenhouse burden, while the sun still radiated significantly less energy than it does today. As methane concentrations fell, less heat remained trapped within the atmosphere. Temperatures began to decline across the planet. On geological timescales, the cooling unfolded rapidly. A powerful feedback mechanism emerged from that initial drop. Ice forming near the poles reflected incoming sunlight back into space. Open water and darker surfaces absorb much of that energy, yet ice behaves differently, acting like a reflective shield. As cooling expanded the ice cover, more sunlight was reflected away. Reduced absorption encouraged further cooling, which produced additional ice. Each step reinforced the next, creating a cycle that strengthened as it spread. Another process intensified the trend. Early oxygen-producing ecosystems continued drawing carbon dioxide from their surroundings through photosynthesis. Carbon dioxide remained an important greenhouse gas even if it was weaker than methane. Its gradual reduction removed another source of atmospheric warming. One greenhouse gas was being chemically dismantled, while another was being steadily consumed by living organisms. Viewed from the perspective of Earth's history, the pace of the transformation was extraordinary. The methane-rich atmosphere that had developed across billions of years was diminishing within a comparatively brief interval. Chemical reactions, biological change, and climate feedbacks were operating together, each amplifying the effects of the others. The cyanobacteria, driving the oxygen increase, remained unaware of the consequences unfolding around them. They possessed no way to sense atmospheric composition on a planetary scale. They could not measure temperature, calculate climate trends, or anticipate the future. Their cells continued capturing sunlight, extracting electrons from water, and releasing oxygen as they always had. Across oceans and shorelines, the same process repeated day after day while Earth's heating system unraveled above them. The decline of methane marked more than an atmospheric transition. It initiated a climatic shift so severe that the world entering it would scarcely resemble the one that had existed before, and the scale of that transformation would soon be written across the entire planet. A layer of stone in northern Canada preserves a remarkable transition. Below it lies evidence of a warmer world shaped by conditions that had endured for immense spans of time. Above it appear the marks of glaciers, debris carried by ice, and sediments deposited in an environment transformed by cold. Between those layers sits one of the most consequential climate shifts in Earth's history. Around 2.4 billion years ago, the Huronian glaciation began. Its duration remains uncertain, though geological evidence suggests it persisted for roughly 300 million to 400 million years. That span exceeds the combined length of all later major ice ages. Entire chapters of biological history would fit comfortably within the interval occupied by this prolonged planetary winter. The reach of the ice was extraordinary. Geologists have identified glacial deposits in regions that once lay near tropical latitudes. These rocks formed far from the poles, indicating that ice advanced far beyond the zones where glaciers typically develop. Conditions that began as cooling at higher latitudes expanded until much of the planet felt their influence. The boundary between polar and tropical climates became increasingly difficult to distinguish. Some researchers propose that the Huronian reached the threshold of a true snowball Earth. In that scenario, oceans were sealed beneath ice extending for kilometers, which is about 0.62 miles in thickness. Global average surface temperatures may have fallen to approximately 50 degrees below 0 degrees Celsius or 58 degrees below 0 degrees Fahrenheit. Once reflective ice covered enough of the surface, sunlight that might otherwise have warmed the planet was redirected back into space, strengthening the cooling already underway. Under those conditions, life faced a world transformed. Surface environments became increasingly hostile, while refuge persisted in a few protected settings. Hydrothermal vent systems on the ocean floor continued releasing heat from Earth's interior. Around these vents, liquid water remained available even when conditions above were dominated by ice. These environments likely served as enduring shelters for portions of the microbial biosphere. Evidence from the Gauganda Formation in what is now Ontario captures part of this history. There, glacial sediments rest directly above much older rocks formed before the freeze. The sequence records a dramatic environmental change within a single geological archive. What had once been a comparatively warm planet entered an era defined by recurring cold. The Huronian was not a single, uninterrupted freeze. Geological records indicate multiple advances and retreats of ice separated by intervals of partial thawing. Volcanic activity continued throughout these cycles, releasing carbon dioxide into the atmosphere. While methane concentrations had collapsed, volcanic emissions gradually accumulated. Across millions of years, that buildup altered the balance between cooling and warming, allowing periodic retreats of the ice. Eventually, geology provided the mechanism that life could not. Volcanoes continued operating regardless of surface conditions, steadily adding greenhouse gases to the atmosphere. As carbon dioxide concentrations rose, enough heat became trapped to weaken the grip of the ice. The thaw emerged from processes deep within the planet itself. The Huronian glaciation revealed an intimate connection between biology, atmosphere, and climate. Microbial activity altered atmospheric chemistry. Atmospheric chemistry reshaped planetary temperature. Planetary temperature transformed the environments in which life struggled to persist. Yet the freeze represented only one dimension of the upheaval, because oxygen itself was continuing a second transformation whose consequences were spreading through every surviving ecosystem. There was no impact crater announcing this disaster, no mountain-sized volcano darkening the sky, no sudden external force arriving from beyond Earth. The agent of destruction was woven directly into the chemistry of a gas that had only recently begun accumulating in the atmosphere. Oxygen, beneficial to much of life today, entered a biosphere that had evolved entirely without it. For organisms shaped by an oxygen-free world, the new gas presented a profound challenge. Oxygen readily participates in chemical reactions that alter organic molecules. Within cells lacking protective mechanisms, it could disrupt enzymes, damage membranes, and trigger chains of reactions capable of harming deoxyribonucleic acid. The molecular machinery that had functioned successfully for billions of years now faced conditions it had never encountered before. The dominant inhabitants of that earlier world included methanogens, sulfur reducing microbes, and numerous anaerobic bacteria occupying ecological roles across oceans, sediments, and shallow coastal environments. Their metabolism depended on chemical pathways developed under permanently oxygen free conditions. No evolutionary history had prepared them for a planet where oxygen was becoming increasingly common. Many lacked the specialized enzymes that later organisms would evolve to neutralize oxygen related damage. As oxygen spread through seawater and eventually the atmosphere, those ancient communities came under growing pressure. Habitats that had remained stable for immense spans of time began changing. Microbial populations that once flourished across broad regions declined. The transformation unfolded across geological timescales, yet its consequences reached nearly every corner of the biosphere. An entire planetary ecosystem entered a prolonged period of collapse. Methanogens were among the hardest hit. Oxygen interfered directly with their biology while simultaneously dismantling the environmental conditions they required. Their habitats shrank as oxygen penetrated more regions of the planet. Their numbers fell as the chemistry supporting methane production became increasingly difficult to maintain. With fewer methanogens surviving, methane output declined further, reinforcing changes already reshaping the climate. What makes this extinction unique is its origin. Later mass extinctions would involve asteroid impacts, volcanic upheavals, shifting continents, and rapid climate disruptions. The oxygen catastrophe emerged from life itself. Cyanobacteria continued carrying out the same metabolic process that had provided a competitive advantage for hundreds of millions of years. The oxygen released as a waste product accumulated until it transformed the environment for nearly every other organism alive. Complete disappearance never occurred. Many anaerobic lineages found refuge where oxygen remained scarce. Deep sediments beneath the seafloor, fractures within subsurface rock, hydrothermal vent systems, and isolated pockets of oxygen-free mud provided sanctuary. In these environments, descendants of those ancient microbes survived while much of the surface world changed around them. Even today, some of their descendants continue similar forms of metabolism. They inhabit locations where oxygen remains limited, preserving biochemical pathways that originated long before the atmosphere acquired its modern character. Their existence offers a living connection to a world that once operated according to very different rules. The true scale of the loss remains difficult to measure. Microbial communities rarely leave the rich fossil record associated with larger organisms. Much of what vanished can only be inferred through chemical signatures preserved in ancient rocks and through the disappearance of once common metabolic patterns. Earth's earliest dominant biosphere faded with little physical trace, yet the story contained another twist. The organisms whose waste product had transformed the atmosphere were not standing outside the consequences of that transformation. The changing world would reach them as well, and the conditions they helped create would impose pressures they had never faced before. A layer of ice can be indifferent to the organism that helped create it. Once frozen conditions spread across large portions of the planet, sunlight struggled to reach the waters where cyanobacteria had flourished for hundreds of millions of years. The same microbes that had transformed atmospheric chemistry now faced an environmental crisis of their own making, though they possessed no awareness of the connection. Oxygenic photosynthesis depended entirely on access to light. When expanding ice covered broad regions of the ocean's surface, that energy supply diminished. Cyanobacteria could continue producing oxygen only where sunlight still penetrated the water. Areas that had once supported thriving microbial communities became increasingly hostile. The biological engine responsible for oxygenation began losing much of its operating space. The rock record preserves traces of this decline. Stromatolites, those layered structures built by generations of microbial mats, become far less abundant during the Huronian glacial interval. Geological formations that once recorded extensive reef building activity shrink dramatically. In certain regions, stromatolite deposits vanish from the record altogether during the coldest phases. Their disappearance reflects ecological contraction on a planetary scale. Atmospheric oxygen did not rise in a smooth upward line through this period. Geochemical evidence suggests repeated fluctuations instead. Intervals of retreating ice open new habitats to sunlight, allowing cyanobacterial populations to expand. Oxygen production increased during those windows. Returning glacial conditions reduced photosynthetic activity once more, pulling oxygen levels downward. The atmosphere shifted between states across spans of millions of years, searching for balance within conditions never previously experienced. Additional complexity emerged from the appearance of organisms capable of using oxygen as a source of energy. These early aerobic life forms consumed oxygen as part of their metabolism. During some intervals, biological consumption increased enough to reduce atmospheric concentrations. Lower oxygen levels permitted temporary recoveries among surviving methane-producing microbes. Methane concentrations rose modestly, contributing to partial warming. Thawing conditions expanded habitable zones for cyanobacteria, encouraging renewed growth and another rise in oxygen production. Earth had entered a prolonged cycle of interacting feedbacks, each influencing the next. Viewed from a wider perspective, the irony becomes striking. Cyanobacteria had finally escaped the long period of suppression imposed by ocean chemistry. Nickel concentrations had fallen, iron sinks had weakened, and oxygen could at last accumulate in meaningful amounts. Their evolutionary success altered the atmosphere, transformed climate, and reshaped the biosphere. Those same achievements pushed their own populations toward severe decline. No villain appears in this story. Natural selection favored organisms capable of exploiting available resources. Cyanobacteria used sunlight and water because those resources offered an advantage. Oxygen emerged as a byproduct of that chemistry. The resulting transformation followed the ordinary rules of evolution and planetary chemistry, neither guided nor restrained by foresight. Many cyanobacterial lineages survived despite the hardships. Small refuges remained where light, liquid water, and suitable environmental conditions persisted. These scattered populations endured through intervals of freezing and thawing, preserving the biological innovation that had already changed Earth forever. The organisms responsible for the first mass extinction, the collapse of the methane world, and the longest glacial episode in planetary history came remarkably close to sharing the fate of the life forms they had displaced. Their survival carried consequences extending far beyond their own lineage. Life endured the harshest conditions Earth had yet produced, and the path by which it endured would shape everything that followed. Survival during the Huronian freeze was never evenly distributed. Life did not remain spread across the planet in the way it had before the cold deepened. It gathered where chemistry, heat, and liquid water still existed, pressed into refuges that could endure conditions the surface could no longer support. Every organism alive today descends from lineages that found some path through that narrowing world. The most reliable refuge lay on the ocean floor. Hydrothermal vents continued drawing heat from Earth's interior, releasing warm fluids and dissolved chemicals into the surrounding water. Even beneath ice that may have extended for kilometers, which is about 0.62 miles, liquid environments persisted around these volcanic systems. Microbes living there did not depend on sunlight. Their energy came from chemical reactions involving minerals, sulfur compounds, and heat, a way of life already familiar from the oxygen-free Earth. Other refuges may have existed closer to the frozen surface. Dark particles of dust and minerals can settle onto ice, absorb sunlight, and melt small pockets downward. These liquid pockets are called cryoconite holes. Modern glaciers in Greenland contain such miniature habitats, and researchers have found microbial communities thriving inside them, including photosynthetic organisms. During the Huronian, similar pockets could have sheltered cyanobacteria within ice, surrounded by meters of frozen water, yet still able to capture enough light to survive. Climate models also suggest that the equatorial ocean may not have frozen completely during every phase. The deep ocean stored heat, and volcanic carbon dioxide continued entering the atmosphere. In some scenarios, narrow belts of open or seasonally thin water remained near the tropics. Such regions would have offered precious access to sunlight, allowing photosynthetic populations to persist when much of the surface ocean was sealed away. Life also retreated into rock, deep within cracks, pores, and mineral surfaces below the seafloor. Microbes capable of drawing energy from chemical reactions could endure without sunlight or surface stability. These organisms, known as chemolithotrophs, used rock chemistry as an energy source. Their world was insulated from ice, atmosphere, and changing weather, connected instead to the slow movement of water through stone. The glaciation acted as a planetary bottleneck. Countless microbial communities that had flourished under earlier conditions disappeared or contracted severely. The survivors were not necessarily the strongest in any simple sense. They were the ones located in the right refuges, carrying metabolisms flexible enough to persist where liquid water, energy, and protection overlapped. That filtering changed the future of life. Populations once spread across broad marine environments became concentrated in fewer habitable zones. Close contact among surviving microbes may have increased opportunities for genetic exchange, competition, and adaptation. The catastrophe narrowed life, then forced what remained into new arrangements. When the ice finally retreated, the survivors entered a planet unlike the one their ancestors had known. Oxygen had become a permanent part of the atmosphere. The old chemical world had fractured. From the refuges that endured the freeze came the lineages that would inherit the transformed Earth, carrying the entire future biosphere through one of the narrowest passages in planetary history. Emerging from the long freeze, Earth carried little chemical resemblance to the world that had existed before oxygen entered the atmosphere. Ice retreated, oceans reopened, and continental surfaces reappeared beneath the sky. The transition marked more than the end of a glacial era. It revealed a planet whose fundamental chemistry had been rewritten. For most of Earth's history, free oxygen had been absent from the atmosphere. Any oxygen produced was rapidly consumed by surrounding materials. Following the Great Oxidation Event, that condition no longer held. Atmospheric oxygen became a persistent component of the air for the first time. Estimates suggest concentrations may have reached somewhere between 1 and 10% of modern levels. By present standards, this remained modest, yet it was sufficient to alter the behavior of nearly every major chemical system on the planet. The continents recorded the change directly. Exposed minerals reacted with oxygen and began forming iron oxides across broad regions. Rocks that had once weathered under reducing conditions entered a different chemical environment. Shades of red, orange, and ochre spread through sediments and exposed surfaces as oxidation advanced. Ancient formations preserved around the world still carry these pigments, visible reminders of the moment Earth's surface chemistry shifted into a new state. The oceans also entered unfamiliar territory. Hundreds of millions of years of rust formation had removed enormous quantities of dissolved iron from seawater. The chemical character of the upper ocean changed as that reservoir diminished. Oxygen could now persist in surface waters instead of vanishing immediately into dissolved metals. New ecological opportunities appeared wherever oxygen remained available for extended periods. Marine environments became more chemically diverse than they had been during the long anoxic ages. Another transformation unfolded above. Methane had dominated greenhouse warming throughout much of early Earth history, yet the rise of oxygen permanently undermined that arrangement. Atmospheric chemistry now favored a world where oxygen remained stable and methane existed only in far smaller quantities. Maintaining oxygen required continual biological production, but once established, the atmosphere no longer reverted to its former condition. The methane world had ended. High above the surface, a second consequence was beginning to take shape. Ultraviolet radiation striking oxygen molecules could separate their atoms. Some of those free atoms combined with intact oxygen molecules, producing ozone. At first, the resulting ozone layer was thin and incomplete, though even a limited shield altered conditions in the upper atmosphere. A barrier against portions of incoming ultraviolet radiation was gradually assembling from the same oxygen that had transformed climate and extinguished countless organisms. This post-glacial Earth still differed greatly from the modern planet. Oxygen remained far below present concentrations. The ozone layer offered only partial protection. Forests, animals, and complex ecosystems lay more than 1 billion years in the future. Even so, the direction of change had become firmly established. Geological processes, atmospheric chemistry, and biological evolution were now interacting within a framework that had never existed before. A new possibility had emerged from that transformed chemistry. Oxygen represented more than an environmental change. It offered access to an energy source far beyond anything available in the older anaerobic world. The door stood open, and some organisms were beginning to cross its threshold. The consequences would reach into every future chapter of life on Earth, reshaping biology from its deepest foundations. A change of this magnitude could never remain merely a shift in atmospheric chemistry. Once oxygen became a persistent feature of the world, life faced a new possibility. The same gas that had frozen climates, transformed oceans, and reshaped ecosystems also carried an opportunity hidden within its reactivity. A few lineages began discovering how to use oxygen itself as part of their metabolism, turning a former threat into a source of extraordinary power. For billions of years, organisms had relied on anaerobic pathways to extract energy from food. Those methods worked, but their limits were strict. A molecule of glucose processed through fermentation yielded only 2 molecules of adenosine triphosphate, the cellular currency that powers biological activity. Aerobic respiration changed that equation dramatically. By combining organic molecules with oxygen through a carefully controlled series of reactions, cells could extract up to 36 to 38 molecules of adenosine triphosphate from the same amount of fuel. The numerical difference appears simple on paper. In practice, it altered what biology could build. Energy places limits on every living system. It determines how much material can be maintained, how rapidly growth can occur, and how many internal processes can operate simultaneously. An organism receiving 18 times more usable energy from the same meal gains access to possibilities unavailable to competitors restricted to fermentation. Cells could invest more resources in repair, regulation, transport, and communication. Larger genomes became easier to maintain. Internal structures could become more specialized. Biological complexity no longer faced the same energetic ceiling that had constrained life throughout the long anaerobic ages. The transformation resembled the difference between a small lantern and an electrical grid. Both provide power, yet the scale of what they can support belongs to entirely different worlds. Many of the traits later associated with complex organisms depend on this energy surplus. Sustained movement, intricate cellular architecture, rapid development, and large collections of cooperating cells all require substantial metabolic income. Aerobic respiration supplied that income. Complexity did not emerge immediately, though the energetic foundation that would eventually support it had appeared. Evidence suggests this transition was prepared long before atmospheric oxygen became widespread. Research tracing the history of oxygen processing enzymes indicates that some microbial populations had already been adapting to localized oxygen-rich environments surrounding photosynthetic communities. Within those pockets, oxygen concentrations could temporarily rise far above surrounding levels. Organisms exposed to such conditions gained opportunities to experiment with biochemical solutions that later became invaluable when oxygen spread across the planet. Natural selection strongly favored those adaptations once oxygen became common. Environments lacking oxygen still existed, though their geographic extent gradually contracted. Regions exposed to oxygen expanded through surface waters, sediments, and coastal habitats. Lineages capable of exploiting oxygen gained access to an energy source continuously replenished by photosynthetic life. Those unable to tolerate it remained confined to shrinking refuges. The world that emerged after the Great Oxidation Event, therefore, offered more than survival. It offered abundance. Aerobic respiration represented the most powerful energy harvesting strategy biology had yet developed, and its consequences would echo through every later chapter of evolution. Even so, greater energy alone could not create the complex cell. One final transformation remained necessary, a biological partnership so improbable that it changed the architecture of life itself. Its beginning came when one cell engulfed another, and neither one died. Among the countless encounters between microorganisms across Earth's early history, one meeting altered the future more profoundly than any climate shift, extinction, or evolutionary competition. Sometime between 2 billion and 1.5 billion years ago, a larger cell surrounded a smaller bacterium and pulled it inside. Such events likely happened many times before and after. On this occasion, however, digestion never reached completion. The captured cell remained alive. The host was probably an archaeon, a member of an old lineage whose ancestors stretched deep into the planet's earliest biological history. The guest was an aerobic bacterium, already equipped with the biochemical machinery needed to use oxygen for respiration. Each possessed abilities the other lacked. One provided a stable internal environment and access to nutrients. The other carried an energy-producing system of exceptional power. Their encounter created a partnership that natural selection steadily refined across millions of years. At first, the relationship may have been fragile. Two formerly independent organisms had to coordinate their activities while sharing the same cellular space. Genes that were once useful for solitary existence became unnecessary. Others gained new value. Generation after generation, functions were exchanged, modified, and integrated. Boundaries that had once separated host from captive became increasingly difficult to define. As the partnership deepened, something unprecedented emerged. The combined cell possessed capabilities unavailable to either participant alone. The archaeal host contributed organizational complexity, while the bacterium supplied abundant energy through aerobic respiration. Together, they could support a level of cellular activity that earlier forms of life could not sustain. Biological possibilities expanded into territory that had previously remained inaccessible. Modern biology preserves evidence of this event in every complex organism alive today. The descendants of that captured bacterium still exist inside cells as mitochondria. They occupy the interior of animals, plants, fungi, and countless microscopic species. Their structure retains traces of an independent past. They reproduce through division. They contain their own genetic material. Their membranes differ from those surrounding other cellular compartments. Each characteristic serves as a reminder that they began as free-living organisms in the distant ocean. The significance of the merger reaches far beyond the origin of a new cell type. Eukaryotic cells, the cells built from this partnership, became the foundation for all visible complexity. Large bodies, specialized tissues, developmental programs, and intricate ecosystems all depend upon the energetic capacity provided by mitochondria. Without that increase in available power, complex multicellular life would face severe constraints. The familiar world of forests, animals, and fungi would never emerge. A long chain of causes stretched behind the event. Aerobic bacteria existed because oxygen had become available. Persistent atmospheric oxygen existed because cyanobacteria transformed planetary chemistry. The oxygen revolution itself followed hundreds of millions of years of chemical and biological change. A sequence that began with microscopic photosynthetic cells ultimately made possible a partnership between two very different organisms. The merger therefore stands as one of evolution's most consequential turning points. A bacterium entered another cell and remained there. Through that improbable outcome, a new kind of life appeared, carrying within it a living record of ancient cooperation. Even now, inside every animal cell, the descendant of that captured bacterium continues its work, preserving a relationship that began in oceans older than any complex organism the Earth would later produce. Deep within the interior of every complex cell, a partnership formed billions of years ago continues without interruption. It unfolds with each heartbeat, each growing leaf, and each dividing cell. The participants are so thoroughly intertwined that they appear inseparable, yet their shared history remains written into the structure of life itself. The aerobic bacterium, once engulfed by an ancestral eukaryotic host, did not fade into the background of evolution. It became the mitochondrion, the energy-producing organelle present in nearly every eukaryotic cell alive today. Although modern mitochondria function as part of larger cells, They retain unmistakable signs of an independent origin. Their existence preserves one of the clearest records of an ancient biological merger. Mitochondria possess their own circular deoxyribonucleic acid, distinct from the deoxyribonucleic acid stored within the nucleus. They reproduce through division, splitting into two descendants in a manner closely resembling free-living bacteria. Their internal machinery differs from that of the surrounding cell. Even the double membrane that encloses them carries the signature of engulfment, a cellular fossil left behind by the moment one organism entered another and remained there. These clues are more than historical curiosities. Modern genetic sequencing allows researchers to compare mitochondrial genes with those of living bacteria. The relationship is measurable and direct. Mitochondria belong within a branch of life connected to proteobacteria, a large group of microorganisms still inhabiting the world today. Their ancestry is therefore preserved in living molecules rather than inferred from distant speculation. A second partnership emerged later in the lineage, leading to plants and algae. On that occasion, a eukaryotic cell engulfed a cyanobacterium. Once again, digestion never reached completion. The captured organism survived and eventually became the chloroplast, the photosynthetic structure responsible for converting sunlight into chemical energy. Chloroplasts retain their own deoxyribonucleic acid, their own bacterial-style ribosomes, and the layered membranes expected from an engulfed cell. This means the two principal energy systems operating within many eukaryotic cells originated as independent organisms. Mitochondria harvest energy through oxygen-based respiration. Chloroplasts capture sunlight, split water, and build organic molecules from carbon dioxide. Each continues performing forms of metabolism that evolved long before plants, animals, or fungi existed. The scale of this inheritance is difficult to grasp. A human body contains roughly 37 trillion cells. Most of those cells contain hundreds or even thousands of mitochondria. The descendants of that single bacterial lineage therefore exist in numbers, reaching into the tens of quadrillions within one person alone. Every breath depends on their activity. Every movement draws upon energy they help generate. Plant life carries an equally deep connection to the distant past. The chloroplasts operating inside leaves today still perform the same core photosynthetic reactions developed by cyanobacterial ancestors billions of years ago. The chain remains unbroken across spans of time that encompass nearly all complex life on Earth. What appears to be a single organism is therefore a community built from ancient unions. Boundaries that once separated partners dissolved through countless generations of shared evolution. Yet the evidence remains embedded within every cell, revealing that cooperation, capture, and coexistence helped shape the living world as profoundly as competition ever did. For nearly 1 billion years, the story of Earth appeared to pause. Mountains rose and wore away. Oceans circulated across the globe. Continents drifted through unfamiliar arrangements. Yet compared with the upheavals that came before and after, this span of time unfolded with unusual steadiness, earning a name that reflects its outward appearance more than its hidden significance, the boring billion. Beginning roughly 1,800,000,000 years ago and extending until about 800 million years ago, the planet settled into a long interval of chemical balance. Atmospheric oxygen persisted as a permanent feature of the world, though at levels estimated to range between 1 and 10% of modern concentrations. That amount was sufficient to support aerobic organisms in sunlit waters, but it remained far below the abundance later associated with large animals and complex ecosystems. The oceans reflected this limitation. Oxygen occupied the upper layers where contact with the atmosphere and photosynthetic activity maintained its presence. Far below, a different chemistry prevailed. Much of the deep ocean remained deprived of oxygen and often rich in sulfur compounds. A vertical boundary separated these environments, creating a stratified world where surface waters and abyssal depths operated under profoundly different conditions. Complex eukaryotic organisms faced restrictions imposed not by competition alone, but by the chemistry surrounding them. Viewed from the perspective of fossils, little seemed to happen. Massive evolutionary radiations were absent. No forests covered the continents. No animals moved across seafloors. Yet the apparent stillness concealed changes occurring within cells. During these long ages, eukaryotic lineages continued refining biological innovations whose consequences would emerge much later. The foundations of future complexity were being assembled piece by piece. Among the most consequential developments was the rise of sexual reproduction. Earlier organisms commonly produced offspring by copying themselves. Sexual reproduction introduced a different strategy, blending genetic material from two parents into new combinations. Each generation became an experiment. Variation accumulated more rapidly, providing evolution with a wider range of possibilities to explore. The pace at which useful traits could appear and spread increased substantially. Evidence also suggests that multicellular organization began appearing in tentative forms. Fossils discovered in rocks from present-day Gabon preserve disc-shaped organisms dating to approximately 2,100,000,000 years ago. These creatures lacked the complexity of later plants or animals, yet they demonstrate that groups of cells had already begun exploring cooperative existence. The path toward larger bodies had opened, even if progress remained constrained. Physics imposed limits that biology could not easily escape. Oxygen must reach cells quickly enough to sustain metabolism. Large bodies require increasing amounts of energy and oxygen delivery. Under the atmospheric conditions prevailing through much of the boring billion, those demands could not be met efficiently. The ceiling on complexity remained firmly in place, holding evolution below thresholds that later organisms would surpass. Near the close of this long interval, geology disturbed the balance. The supercontinent Rodinia began breaking apart around 800 million years ago. Continental fragments shifted, mountain ranges formed, and weathering intensified. Rivers carried fresh nutrients into the oceans, altering chemical cycles that had persisted for immense spans of time. Stability gave way to transformation. The world that emerged from this transition was preparing for another oxygen increase, one that would reshape both ocean and atmosphere. A new chapter in planetary chemistry was beginning to unfold. A continent breaking apart may seem an unlikely beginning for a biological revolution, yet the second great rise of oxygen began with stone. Around 800 million years ago, the supercontinent Rodinia entered a prolonged phase of fragmentation. Land masses separated, mountain belts lifted skyward, and fresh rock surfaces spread across immense regions of the planet. Those geological changes altered the chemistry of Earth in ways that would echo through every future ecosystem. Rain, rivers, and weathering gradually wore down the newly exposed terrain. Minerals released from the rocks traveled toward the oceans, carrying phosphorus and other nutrients that had previously been locked within continental crust. Surface waters responded quickly. Photosynthetic organisms multiplied across broad marine regions, converting sunlight into organic matter at rates far above those of earlier ages. The significance of that productivity lay not only in oxygen production, but also in burial. Ordinarily, much organic material is broken down after death, consuming oxygen and returning carbon dioxide to the environment. During this interval, larger quantities of organic carbon became trapped within accumulating seafloor sediments before decomposition could complete the cycle. Each portion buried and isolated from the atmosphere altered the balance. Carbon remained underground while oxygen stayed behind. Across millions of years, that imbalance accumulated. Atmospheric oxygen climbed in a stepwise progression rather than a single, dramatic leap. Evidence of the change survives in ancient rocks. Variations in chromium, uranium, and molybdenum, preserved within sediments, tell the same story through independent chemical pathways. Though each element records environmental conditions differently, their signals converge on a common conclusion. Earth was entering a more oxygen-rich state than it had known during the long ages that followed the first oxygenation event. The oceans transformed alongside the atmosphere. For much of the preceding billion years, oxygen had occupied only upper waters, while deeper regions remained chemically restrictive. As oxygen concentrations increased, circulation gradually carried it farther downward. At last, substantial portions of the seafloor became hospitable to complex organisms. Habitats that had remained inaccessible for immense spans of time opened for occupation. The architecture of marine ecosystems gained room to expand. Energy availability expanded with it. Aerobic organisms already possessed a powerful metabolic system, yet its potential had been constrained by limited environmental oxygen. Higher concentrations allowed larger cells and more active lifestyles. Biological structures that consumed significant energy could now be maintained more effectively. The ceiling that had restricted complexity for so long began lifting, creating opportunities unavailable during the boring billion. The path was neither smooth nor stable. During this broader interval, Earth experienced severe glaciations, including the Sturtian and Muranoan events. Ice advanced across large portions of the planet, disrupting ocean circulation and altering nutrient flows. Geological records suggest these episodes may have contributed indirectly to further oxygenation by influencing patterns of carbon burial. Once again, planetary upheaval and biological opportunity became linked through chemistry. As oxygen continued accumulating, its influence extended far above the oceans. High in the atmosphere, oxygen molecules interacted with incoming radiation and with one another, generating increasing quantities of ozone, a protective layer slowly assembled overhead. The process unfolded gradually, yet its consequences would prove profound. For billions of years, the exposed surface of the continents had remained a hostile place for complex organisms. The obstacle was not temperature, nor the absence of water alone. It arrived continually from above, carried in powerful ultraviolet radiation that struck the ground without restraint. The sky itself was beginning to change that reality. Above the oceans, a transformation was unfolding that no organism could see. High in the atmosphere, far beyond the reach of waves, currents, and seafloor sediments, oxygen was beginning to reshape the relationship between Earth and the Sunday. The consequences would eventually extend to every forest, every animal, and every footprint ever left on dry ground. For most of planetary history, the continents existed as biologically hostile terrain. The obstacle was neither extreme heat nor a lack of suitable minerals. It came from ultraviolet radiation streaming downward through an atmosphere unable to stop it. Exposed cells faced continual molecular damage. Deoxyribonucleic acid absorbed the energy directly, proteins were altered, and living tissues suffered disruption faster than most organisms could repair it. Any attempt to establish complex ecosystems on open land encountered a barrier imposed by physics itself. Water provided refuge from that threat. Oceans, lakes, and shallow coastal environments absorbed large portions of incoming ultraviolet radiation before it could reach living organisms. Life therefore remained tied to aquatic settings for billions of years. The shoreline was less a frontier than a boundary separating protected environments from a surface world that offered little biological safety. The mechanism that changed this situation emerged from oxygen itself. In the upper atmosphere, ultraviolet radiation struck ordinary oxygen molecules and split them apart. Individual oxygen atoms then collided with intact oxygen molecules, creating a new form composed of three oxygen atoms. This substance, ozone, accumulated gradually in atmospheric layers, where these reactions occurred most efficiently. At first, the effect was limited. Oxygen concentrations remained too low to support a substantial ozone shield. As the Neoproterozoic oxygenation event progressed, however, atmospheric oxygen continued increasing. Additional oxygen supplied more raw material for ozone formation. Across immense spans of time, the protective layer strengthened. Radiation that once traveled directly to the ground encountered an increasingly effective filter before reaching the surface. By the closing chapters of the Precambrian, around 541 million years ago, the atmosphere had changed enough that large portions of harmful ultraviolet radiation were intercepted before arrival at the continents. Land did not become completely safe, yet it became survivable in a way it had never been before. Conditions that had excluded complex organisms for billions of years were beginning to relax. The chain of cause and effect stretched across extraordinary distances in time. Cyanobacteria released oxygen as a metabolic byproduct. Oxygen accumulated through geological ages. Atmospheric reactions converted a fraction of that oxygen into ozone. Ozone reduced ultraviolet radiation reaching the surface. Through that sequence, organisms inhabiting shallow waters altered environments they would never occupy and prepared habitats they would never witness. Every terrestrial ecosystem traces its existence through that pathway. Plants rooted in soil, insects crossing leaves, reptiles warming on rocks, mammals moving across grasslands, and human beings building cities all depend upon atmospheric protection assembled from oxygen. The shield overhead is so familiar that its presence often escapes notice, yet without it, the continents would remain largely inaccessible to complex, exposed life. The remarkable aspect of the process lies in its lack of intention. The microorganisms responsible possessed no awareness of continents waiting beyond the horizon of time. They neither planned nor predicted the consequences of their chemistry. Through countless generations, they altered the atmosphere molecule by molecule. With oxygen rising, deep oceans becoming more hospitable, and ultraviolet radiation increasingly restrained, the conditions required for complex animals were finally approaching completion. The stage was ready for an expansion of life, unlike anything Earth had experienced before. A span of 25 million years can seem immense when measured against a human life. Against the age of Earth, it is scarcely a moment. Within that brief interval, beginning around 541 million years ago, life produced an array of forms so diverse that nearly every major animal blueprint alive today can trace its roots to that burst of innovation. The stage had been prepared during the Ediacaran period, Across seafloors enriched by rising oxygen, unusual multicellular organisms spread through shallow marine environments. Some resembled fronds anchored to the sediment. Others appeared as disks, ridges, or quilted shapes, unlike anything familiar today. Their bodies lacked shells, teeth, and bones. Many left delicate impressions preserved in fine-grained sediments, records of communities occupying ecological space that had only recently become available. A number reached 1 meter in length, which is about 3.3 miles. Their world was still comparatively simple, though the foundations of something larger were already taking shape. Then the pace changed. Geological layers marking the Cambrian reveal an expansion of biological complexity that transformed marine ecosystems. Hard external coverings appeared, skeletons reinforced bodies. Limbs became jointed. Burrowing animals altered sediments from within. Predators pursued prey through open water. Nervous systems grew more sophisticated, supporting behavior that would have been unattainable in earlier eras. The oceans began to resemble living systems recognizable in principle today. Among the most celebrated windows into this period is the Burgess Shale in what is now British Columbia. Preserved, there are organisms whose soft tissues survived fossilization with unusual fidelity. Anomalocaris moved through Cambrian waters as a leading predator, reaching roughly half a meter in length. Opabinia carried 5 eyes and a flexible frontal structure unlike anything in modern oceans. Hallucigenia puzzled generations of researchers because its anatomy appeared so unfamiliar that early reconstructions placed parts of its body in the wrong orientation. These creatures reflected a world testing possibilities on a remarkable scale. Rising oxygen lay at the center of this transformation. Cells, powered by aerobic respiration, possessed access to energy levels unavailable during earlier chapters of planetary history. Larger bodies became sustainable. Muscle tissue could support prolonged activity. Complex sensory organs became feasible. Neural networks capable of processing increasing amounts of information could be maintained. Biological architecture expanded because the energetic budget supporting it expanded as well. Eyes illustrate this connection particularly well. Vision demands continuous energy investment. Once visual predators emerged, prey species faced new selective pressures. Protective coverings, improved movement, defensive structures, and behavioral adaptations spread through populations. Evolutionary change accelerated as organisms responded to one another. Ecological relationships multiplied, generating a network of interactions far richer than those of previous ages. The Cambrian explosion was therefore more than a sudden appearance of strange animals. It represented life-exploring opportunities created through billions of years of atmospheric and biological change. Oxygen had lifted a long-standing ceiling on complexity, and living systems responded with extraordinary speed. Body plans, feeding strategies, sensory systems, and ecological roles diversified across oceans that had become increasingly hospitable to experimentation. From the first cyanobacteria releasing oxygen into the environment, to the arrival of eyes, shells, predators, and mobile animals, a chain of consequences stretched across roughly 2,400,000,000 years. In the Cambrian, that long, invisible story finally became visible in stone. Inside every breath, the ancient story of oxygen becomes immediate. Air enters the lungs, oxygen crosses into the blood, and the bloodstream carries it towards cells that depend upon structures older than animals, older than plants, and older than the Cambrian seas. Those structures are mitochondria, descendants of bacteria captured between 1.5 and 2 billion years ago. Mitochondria are not inventions built from scratch by animal cells. They are inherited partners with a history of their own. In human cells, and in the cells of nearly every animal alive, they still carry circular deoxyribonucleic acid, separate from the deoxyribonucleic acid stored in the nucleus. They still divide in a way that resembles free-living bacteria, splitting into two. Their double membranes preserve evidence of an engulfment event so old that it predates the bodies they now help power. When oxygen reaches a mitochondrion, it enters one of biology's most important energy systems. Electrons move through a carefully arranged chain of molecular steps. At the end of that chain, oxygen accepts those electrons and combines with hydrogen to form water. The movement of energy through this system helps drive a molecular turbine known as adenosine triphosphate synthase, which can rotate at roughly 9,000 revolutions per minute. The product is adenosine triphosphate, the energy currency used by complex life. Every muscle contraction, every nerve impulse, every protein assembled, and every thought formed in the human brain requires energy supplied through this chemistry. Animal life runs on controlled oxygen use inside cellular structures descended from once independent bacteria. The catastrophe that poisoned the early world eventually became the process sustaining consciousness. The organisms that began the oxygen story remain alive as well. Cyanobacteria still inhabit oceans, lakes, soils, hot springs, rock surfaces, and polar environments. Their lineage has persisted across nearly 3 billion years, surviving the glaciations, extinctions, and atmospheric revolutions connected to their own chemistry. In many places, they still split water, use sunlight, build sugars, and release oxygen into the world around them. Among the most important modern cyanobacteria is Prochlorococcus, a marine genus discovered only in 1986. These cells are among the most abundant photosynthetic organisms known, with estimates reaching around 3 octillion individuals in the global ocean. Prochlorococcus may contribute roughly 20% of Earth's oxygen production, a share greater than that of all tropical rainforests combined. Their size makes that influence even more striking. A single Prochlorococcus cell measures about 0.6 micrometers across. Hundreds could fit across the width of a human hair. They remained unknown to science until the late 20th century, yet their collective activity helps maintain the atmosphere every breathing organism depends upon. The story of oxygen is therefore not confined to rocks, oceans, or distant ages. It is present inside the body. The mitochondria in human cells belong to it. The oxygen entering the lungs belongs to it. The chemistry, keeping a heart beating through the night, descends from reactions that began in shallow waters billions of years ago, when microscopic life released a waste product that would one day make thought possible. Across the full arc of oxygen's history, the chain begins to look less like a straight road and more like a narrow passage through many possible worlds. Each stage depended on conditions that could have shifted slightly and produced a different planet. Earth became oxygenated, complex, and eventually conscious, yet the path leading there was built from events with no guarantee of repetition. The first requirement was oxygenic photosynthesis. Photosystem 2, the molecular machinery able to split water and release oxygen, appears to have evolved only once in Earth's history. Its emergence depended on a specific arrangement of proteins, pigments, and metal centers working together with extraordinary precision. If that arrangement had never appeared, life might still have flourished, but it would have remained bound to older chemistries that did not fill the atmosphere with oxygen. The second requirement lay in the ocean. For hundreds of millions of years, dissolved iron absorbed the oxygen cyanobacteria produced. A planet with a deeper iron reservoir, a more iron-rich mantle, or more persistent volcanic supply could have prolonged that trap far beyond Earth's timeline. Oxygen might have remained buried as mineral rust, never reaching the sky in meaningful amounts. Nickel added another layer of contingency. Early concentrations helped suppress cyanobacterial expansion while supporting methane-producing microbes. If nickel had stayed high for longer, photosynthetic populations might have remained limited even with water and sunlight available. The biological engine of oxygen production would have run below the threshold needed to overcome the planet's chemical sinks. Survival also mattered. Cyanobacteria caused transformations that nearly destroyed many of their own habitats. Ice, toxic oxygen, methane collapse, and ecological disruption could have reduced them beyond recovery. Their persistence through the catastrophe was not a planned outcome. It was the result of refuges, adaptation, and chance distributed across a changing planet. A billion years later, geology intervened again. The breakup of Rodinia altered weathering, nutrient delivery, carbon burial, and oxygen levels. Without that continental fragmentation, the second oxygen surge might have been delayed or weakened. The oxygen needed for large animal bodies may have remained out of reach for far longer. Scientists still disagree about what this pattern means beyond Earth. Some argue that oxygenic photosynthesis offers such a powerful advantage that it may eventually arise on many water-rich rocky planets. Others emphasize the apparent singularity of photosystem II and the many geological conditions required for oxygen to persist. Both views remain possible because Earth is still the only inhabited planet whose deep history can be read in detail. The difference matters. If oxygen-producing life is rare, the universe may contain many living worlds that never announce themselves through oxygen-rich skies. They could be microbial, active, and chemically complex, yet invisible to the searches designed around atmospheres like ours. If oxygenation is common, then worlds with animals, eyes, movement, and ecological complexity may be one chapter in a broader pattern. For now, Earth's story stands alone. Its oxygen was neither obviously inevitable nor impossibly accidental. It emerged from a sequence of chemistry, geology, survival, and chance. That uncertainty now reaches outward, toward planets around other stars, where scientists search for signs that the same threshold may have been crossed somewhere else. A molecule drifting through a distant atmosphere can carry a story across light years. When astronomers study worlds orbiting other stars, they are often searching for chemical traces that reveal processes hidden far below the clouds. Among all the gases known to science, oxygen occupies a special place because Earth's own history demonstrated how difficult it is to accumulate and how much effort nature must expend to keep it present. Oxygen is a restless participant in planetary chemistry. It reacts with exposed minerals, combines with volcanic gases, and becomes locked into countless compounds. Left without replenishment, atmospheric oxygen gradually disappears as these reactions consume it. On Earth, maintaining a substantial oxygen atmosphere requires a continuous source. For billions of years, that source has been life. The lesson emerging from Earth's deep past is straightforward and powerful. Large quantities of atmospheric oxygen are far easier to explain with biology than without it. This realization transformed the search for life beyond Earth. Oxygen became what scientists call a biosignature, a chemical clue that may point toward living systems. Detecting oxygen in the atmosphere of a distant planet would suggest that something is actively replacing the gas as quickly as chemistry removes it. Such a discovery would stand among the most significant scientific findings ever made, connecting a remote world to the same biological processes that reshaped Earth. Modern telescopes pursue this possibility by studying starlight that passes through planetary atmospheres. As a planet crosses in front of its star, tiny fractions of light filter through layers of gas before continuing toward Earth. Different molecules absorb specific wavelengths, leaving identifiable patterns within the spectrum. By examining those patterns, researchers can infer the presence of gases that cannot be observed directly. Oxygen and ozone remain among the most important targets in these investigations because of the role they played in Earth's transformation. Yet Earth's own story introduces caution. Cyanobacteria were producing oxygen long before atmospheric oxygen became abundant. For hundreds of millions of years, the gas disappeared into oceans rich in dissolved iron and into countless chemical sinks scattered throughout the environment. Life was already altering the planet, but a distant observer would have seen little evidence of that activity in the atmosphere. The biosphere existed while its signature remained concealed. That possibility extends naturally to other worlds. A planet could host thriving photosynthetic organisms and still appear oxygen poor if its oceans, crust, or mantle continue absorbing the oxygen being produced. Geological conditions matter as much as biological ones. Differences in iron abundance, volcanic activity, ocean chemistry, or nutrient cycles could delay atmospheric oxygenation for immense spans of time. A living world might therefore resemble a lifeless one when viewed through a telescope. For this reason, the search for oxygen is also a search for planetary timing. Detecting oxygen may indicate that a world has crossed a threshold similar to the one Earth crossed long ago. Failing to detect oxygen does not close the case. It may only mean that the planet occupies an earlier chapter in its own history. Each observation gathered by the James Webb Space Telescope adds another piece to that larger puzzle. Every atmospheric spectrum invites comparison with the long path Earth followed. Somewhere among those distant worlds, life may already be reshaping a planet in ways that remain invisible from afar, just as cyanobacteria once transformed Earth while leaving almost no trace in the sky above them. Among all the actors that have moved through this story, one group stands apart through sheer persistence. Continents assembled and broke apart. Oceans changed chemistry. Ice advanced across much of the planet and later retreated. Entire branches of life appeared, flourished, and vanished. Through each transformation, cyanobacteria endured, continuing a way of life that began long before animals, forests, or even complex cells entered the world. Their presence today spans nearly every environment where light and moisture can be found. Cyanobacteria drift through the upper layers of the ocean, spread across freshwater lakes, cling to soil crusts, inhabit polar ice, and grow in hot springs whose conditions would challenge many other organisms. Some live inside translucent stones in deserts, where rainfall is scarce and sunlight arrives, filtered through rock. Across habitats that seem unrelated, the same basic biological strategy persists, converting light into chemical energy while releasing oxygen as a byproduct. What makes this endurance especially striking is how little their central machinery has changed. Molecular studies of living cyanobacteria reveal that photosystem II, the remarkable protein complex responsible for splitting water and releasing oxygen, remains fundamentally similar to the version that emerged billions of years ago. Evolution has modified details, refined regulation, and adjusted supporting systems, yet the core reaction remains recognizable across deep time. A mechanism that altered the planet proved so effective that natural selection never replaced it. The atmosphere itself reflects that success. Cyanobacteria continue to contribute directly to global oxygen production, while their descendants inside plant and algal chloroplasts generate much of the remainder. Every oxygen-rich breath depends on photosynthetic systems whose ancestry traces back to those distant microbial innovators. The relationship between modern life and cyanobacteria is therefore immediate rather than historical. Their activity is woven into the chemistry of every day. One species group illustrates this influence particularly well. Prochlorococcus, identified only in the late 20th century, occupies enormous regions of the open ocean. Individual cells are extraordinarily small, measuring roughly zero point 0.6 micrometers across. Hundreds could fit across the width of a single human hair. Despite their size, their collective abundance is staggering. Estimates suggest a global population of roughly 3 times 10 to the 27th power individual cells, making them among the most numerous organisms ever known. Together, they contribute a substantial fraction of the oxygen entering Earth's atmosphere. Their record of survival is equally impressive. Cyanobacteria persisted through the oxygen crisis they helped create. They endured global glaciations, atmospheric upheavals, volcanic episodes, asteroid impacts, and mass extinctions that reshaped the biosphere. Many celebrated groups of organisms occupied Earth for only a brief interval by comparison. Cyanobacteria remained present before those lineages appeared and continued after they disappeared. The cells drifting through the ocean tonight participate in a chain without interruption. Each generation connects to earlier generations through an unbroken sequence of reproduction extending back across billions of years. While dinosaurs rose and vanished, while mammals diversified, while human civilizations emerged, cyanobacteria continued harvesting sunlight and releasing oxygen into the world around them. Seen from that perspective, their story carries a different kind of significance. They are neither relics nor survivors, clinging to the margins of history. They remain active architects of the planetary environment, performing the same transformative chemistry that altered Earth's destiny. The atmosphere surrounding every living creature is, in part, a continuing expression of their work. Seen from the distance of deep time, oxygen's rise carries a meaning larger than chemistry alone. It became evidence for one of the most unsettling interpretations of Earth's living history, the Medea hypothesis, proposed by paleontologist Peter Ward. The name comes from Greek mythology, where Medea destroyed her own children. Applied to Earth, the idea suggests that life can damage the very systems that allow it to survive. A more familiar view, the Gaia hypothesis, proposed by James Lovelock, imagines life as a stabilizing force. In that picture, the biosphere tends to regulate climate and chemistry in ways that maintain habitability. The Medea hypothesis turns that image around. It argues that life often destabilizes planetary systems, changing air, oceans, and climate through ordinary biological activity until the world becomes dangerous for many of the organisms living within it. Cyanobacteria provide the clearest example. They released oxygen as waste while pursuing their own survival. That waste dismantled the methane greenhouse, poisoned the dominant anaerobic biosphere, helped trigger the longest glacial episode in Earth's history, and pushed cyanobacteria themselves into crisis when ice blocked the sunlight they needed. No intention was involved. The catastrophe grew from successful metabolism repeated across immense time. The same pattern has appeared in later chapters. During the Devonian period, around 400 million years ago, land plants spread across the continents and transformed rock weathering. Their roots broke minerals apart. Their growth drew carbon dioxide from the atmosphere, and their burial altered carbon cycles. Forests helped cool the planet, contributing to environmental stress associated with the late Devonian mass extinction. In a different world, plants affected carbon dioxide, much as cyanobacteria had affected methane. The mechanism behind this pattern is rooted in natural selection. Evolution favors traits that help organisms survive and reproduce within immediate conditions. It does not reward foresight across geological time. A lineage can benefit from producing a waste product, altering a nutrient cycle, or transforming an atmosphere, while the long-term consequences remain invisible to the pressures shaping its biology. An organism releasing a harmful byproduct does not automatically stop when that byproduct accumulates. If the process improves survival in the present, natural selection may refine it further. The surrounding environment must then absorb the waste, adapt around it, or be transformed by it. The Great Oxidation Event showed this principle at planetary scale, with oxygen turning from microbial exhaust into a force that remade Earth. Earth endured because the collapse was incomplete. Some organisms survived in refuges, others evolved ways to use oxygen. Cyanobacteria persisted through the consequences of their own success. The New World was assembled from survivors, accidents, and chemical leftovers, then inherited by lineages able to live within conditions the old biosphere could not withstand. The Medea hypothesis does not require villains. It describes a pattern in which life, acting only through ordinary survival, can reshape a planet beyond recognition. Oxygen's history leaves that pattern written across stone, atmosphere, and living cells, bringing the story toward a deeper question about consequence without intention and change without any organism knowing the scale of what it had done. Imagine compressing the entire journey into a single thread and tracing it backward through time. Every transformation described across 2,700,000,000 years, every extinction, every frozen ocean, every complex cell, every animal body, every forest, every mind capable of reflection, can be followed to one biological innovation that emerged within a lineage of microscopic organisms inhabiting an early world. The reaction at the center of that story was oxygenic photosynthesis. It did not appear as part of a plan. No future stood before the organisms that evolved it. No mechanism existed to anticipate oceans changing, atmospheres shifting, or ecosystems collapsing and rebuilding. The reaction persisted because it offered an advantage in a particular environment, helping individual cells gather energy and survive competition for light and nutrients. Everything beyond that immediate benefit lay outside the reach of evolution's sight. Natural selection works only within the present. It favors traits that improve survival and reproduction under existing conditions. Future consequences do not influence the process. Cyanobacteria were never selected to transform a planet. They were selected to harvest energy efficiently. Atmospheric oxygen, global glaciation, and the eventual rise of complex life emerged as consequences extending across spans of time so immense that no living organism could perceive them. Viewed from that perspective, the history of complex life rests upon contingency. The word does not imply error or malfunction. It describes outcomes that were possible, yet never guaranteed. If oxygenic photosynthesis had evolved differently, if the chemistry of the oceans had trapped oxygen indefinitely, or if geological conditions had shifted along another path, Earth would still possess life. Microbial ecosystems might cover the planet in extraordinary variety. Metabolisms unknown today could flourish in seas and sediments. The world would remain inhabited, though without animals, forests, or any familiar form of complexity. That realization extends beyond Earth itself. A planet sharing similar oceans, continents, and biological ingredients could experience an entirely different history. The chain connecting microbial competition to atmospheric oxygen involved many linked events, each dependent on circumstances that might have unfolded another way. Repeating the beginning would not guarantee the same ending. The route from simple cells to complex organisms appears less like a straight road and more like a branching landscape filled with alternatives. Consider the oxygen entering a pair of lungs tonight. Its presence depends on countless events layered across deep time. The mitochondria within human cells still rely on that oxygen. Nerve cells use it to sustain electrical activity. Muscles use it to move. Thought itself depends upon chemical processes powered by a partnership forged long before animals appeared. The chain stretches uninterrupted from ancient microbial communities to every conscious moment occurring now. There is a particular weight in recognizing that none of it was directed toward a goal. The biosphere was not assembling future creatures. Cyanobacteria were not preparing the way for plants, animals, or human beings. Catastrophe and opportunity remained intertwined throughout the process. Survival belonged to whatever could persist within changing conditions, carrying forward pieces of older worlds into newer ones. The perspective that emerges is neither one of design nor of meaninglessness. It is the recognition that blind processes produced consequences of extraordinary scale. Across a brief interval of geological time, consciousness appeared and gained the ability to look backward along the entire chain, seeing for the first time how a planetary transformation became a living world capable of understanding its own origin. Take the entire timeline and stretch it across the mind at once. At one end stands a cyanobacterial cell drifting through a distant ocean, releasing oxygen while splitting water molecules for energy. At the other stands a living human being drawing a breath. Between those moments lies roughly 2,700,000,000 years, a duration so immense that ordinary intuition struggles to hold it, yet every breath carries evidence that the connection is real. Across that span, Earth transformed repeatedly. Continents gathered into supercontinents and later fragmented. Mountain ranges rose and wore away. The sun itself grew brighter, increasing its energy output by roughly 25% compared with the world inhabited by the earliest cyanobacteria. Oceans changed their chemistry. Atmospheres changed their composition. Entire ecosystems appeared, flourished, and vanished. Through each transformation, oxygen followed a winding path from absence to abundance. Its concentration did not climb in a straight line. Following its early rise, oxygen lingered at comparatively low levels for immense stretches of time. Later increases opened new ecological possibilities. During the Carboniferous period, around 300 million years ago, atmospheric oxygen may have approached 35%. That world differed noticeably from the one familiar today. Fires ignited more readily and traveled through coal swamp forests with greater intensity. Layers of fossil charcoal preserved in ancient rocks, record landscapes where combustion was a frequent participant in ecological change. The same atmospheric richness helps support creatures that appear almost improbable by modern standards. Insects rely on networks of air-filled tubes rather than lungs. Under higher oxygen conditions, That method of respiration could sustain larger bodies. Meganeura, a predator resembling a dragonfly, reached wingspans of approximately 70 centimeters. Arthropleura, a many-legged arthropod, grew to lengths approaching 2.5 meters. Such forms belong to a particular atmospheric chapter, one written by oxygen levels exceeding those of the present world. Throughout every geological rearrangement, one process endured. Cyanobacteria continued capturing sunlight. The molecular machinery responsible for splitting water and releasing oxygen remained fundamentally effective. Natural selection refined details across billions of years, yet the core reaction persisted. It survived continental migrations, climatic upheavals, asteroid impacts, and mass extinctions. Many famous lineages entered the story and disappeared from it, while that chemistry continued uninterrupted. The atmosphere containing about 21% oxygen today is not a permanent setting fixed into the planet. It exists as a balance maintained by living systems and geological cycles. Photosynthesis adds oxygen. Respiration, decay, weathering, and fire remove it. The concentration remains stable because those processes presently offset one another. Earth has occupied different balances before, and the rocks preserve evidence of each one. Looking across the full sweep of oxygen's history, continuity becomes the defining feature. Catastrophes matter, evolutionary innovations matter. Yet beneath those dramatic changes runs a thread connecting distant ages to the present. A biochemical solution discovered by microorganisms in a remote world never ceased operating. The reaction continued through every chapter that followed. Tonight it remains active in oceans, lakes, soils, and countless microscopic communities. It persists without awareness of the worlds it helped create. The oxygen entering a sleeping body traces back through that unbroken chain, linking a modern breath to one of the oldest living processes on Earth, and carrying the story toward a final reflection on what it means that a creature eventually emerged, capable of noticing the connection. Across all the ages described in this story, one possibility remains suspended beyond the horizon of certainty. A lineage of microorganisms altered a planet, transformed its atmosphere, reshaped evolution, and eventually produced creatures capable of reconstructing the entire sequence from fragments preserved in stone. The question that remains is whether this has happened anywhere else. The observable universe contains an almost incomprehensible number of stars, commonly estimated at around 200 billion trillion. Around many of those stars orbit planets. Some are likely rocky. Some may possess liquid water. A fraction may host chemistry capable of sustaining life. Yet the abundance of worlds does not automatically answer the deeper problem. The probability of the particular chain that led from oxygen-producing microbes to complex animals remains unknown. Current scientific estimates stretch across an enormous range because there is only one example available for study. At the center of the uncertainty sits a molecular innovation that changed everything. Photosystem 2 appears to have emerged only once in the known history of Earth. No alternative pathway has been discovered that produces atmospheric oxygen on a comparable scale. If that biochemical architecture represents an exceptionally improbable event, then many inhabited planets may remain locked in microbial stages for billions of years. Such worlds could teem with living organisms while leaving no atmospheric signal obvious enough for distant observers to detect. Another possibility remains equally plausible. Biology operating for sufficient time on a suitable planet may repeatedly discover oxygenic photosynthesis because the energetic rewards are so substantial. In that view, oxygen rich atmospheres may arise again and again throughout the cosmos. Eyes, nervous systems, active predators, and ecological complexity could emerge as recurring chapters rather than singular events. The history reconstructed from Earth's rocks would then represent one example of a broader pattern written across many worlds. Imagine the significance of detecting atmospheric oxygen around another planet. Such a discovery would reveal that the chain unfolded at least twice. Somewhere, beyond interstellar distances, a biosphere would have crossed its own threshold. Its details would differ. Its species would be unfamiliar. Its history would belong to another world entirely. Yet the fundamental transformation from a low oxygen planet to an oxygen rich one would no longer be unique. A second origin would carry profound implications for the prevalence of life throughout the universe. An opposite result would be equally meaningful. As telescopes improve and surveys expand, a continuing absence of oxygen signatures would gradually narrow the possibilities. Each empty spectrum would suggest that the pathway leading to atmospheric oxygen may be rarer than hoped. In that scenario, the air surrounding Earth would become more extraordinary, representing an outcome achieved by very few planets. Modern observatories already pursue this search. The James Webb Space Telescope examines distant atmospheres through the faint fingerprints embedded in starlight. Future instruments will extend that effort with greater sensitivity. Molecules suspended around remote planets leave traces in the light passing through them, carrying information across distances so immense that travel remains unimaginable. Whether oxygen proves common or scarce, the answer will alter humanity's understanding of its place in nature. Every observation adds another piece to the puzzle. And as this search continues, each breath serves as a reminder that 2,700,000,000 years of consequences remain present in the atmosphere, connecting the question of other worlds to the air sustaining life here tonight. Set aside the numbers for a moment. Let the timelines recede. Let the continents, ice ages, extinctions, oceans, and drifting worlds settle into the background. What remains at the center of this story is something almost impossibly small, a microscopic organism floating in a distant ocean billions of years ago, carrying out a chemical reaction that would eventually alter the fate of an entire planet. That organism was not pursuing a grand objective. It was gathering energy from sunlight, separating water into its components, assembling the molecules needed to survive another cycle of growth and division. Oxygen emerged from the reaction as excess material, released into the surrounding environment because there was nowhere else for it to go. The cell could not perceive the atmosphere. It could not anticipate geological change. It possessed no awareness of consequences extending beyond the next moment of existence. Yet, the discarded molecule accumulated. It transformed oceans. It altered the chemistry of the sky. It extinguished forms of life that had dominated Earth for immense spans of time. It helped drive a planetary freeze. It opened pathways for new metabolisms. It enabled the rise of larger cells, more intricate bodies, active movement, complex ecosystems, and eventually minds capable of asking how any of it happened. Seen from the perspective of deep time, oxygen became many things at once. It was a toxin and an opportunity. It was destruction followed by possibility. It reshaped the conditions under which evolution operated, changing the range of biological experiments available to life. Every turning point described across this journey traces through that transformation, linked by a chain of consequences, stretching across 2,700,000,000 years. Inside the body tonight, that chain remains active. Oxygen enters the lungs and passes into the bloodstream. From there, it travels to cells containing mitochondria, descendants of bacteria that entered into partnership long before animals existed. Within those structures, oxygen participates in reactions that release energy from food. The process powers muscles, supports organs, maintains circulation, and sustains the electrical activity of the brain. Each moment of awareness depends upon chemistry rooted in events unimaginably distant in time. Beyond the body, cyanobacteria continue their work. They occupy oceans, lakes, soils, rocks, and environments scattered across the planet. Their molecular machinery remains strikingly similar to the machinery that operated in remote ages before forests, before animals, before complex cells transformed the world. Generation after generation, the reaction continues. Water is divided, energy is captured, oxygen is released. There is a particular weight in recognizing that the most transformative development in Earth's history emerged without foresight. No organism intended the outcome. No guiding process aimed toward consciousness, civilization, or understanding. A biochemical innovation spread because it functioned effectively for the organisms carrying it. Everything else followed from consequences, unfolding across spans of time, far beyond any individual life. And so the story arrives here, at a breath drawn in the present. The oxygen entering that breath belongs to a process older than animals, older than forests, older than nearly every familiar feature of the living world. The extraordinary part is not merely that the chain endured. The extraordinary part is that, after billions of years of consequences, something now exists that can trace the entire path backward, from a single microscopic reaction to the awareness contemplating it before sleep.